Great. Well, thanks, Alan, Samantha, and Kresha for organizing this great workshop. If you read my title and abstract, I've been so inspired by this week of talks that I decided to actually pivot and talk about very new stuff. So you'll see that this title is a little bit different than what you read, but I will touch on a little bit of what was in that original abstract before jumping into brand new stuff that I've been working on. So a quick kind of overview of what I plan on talking about. I want to talk a little bit about my mindset in terms of approaching these types of problems, and very much the loop of the being inspired by biology, doing the math, and then looping back to the biology before then jumping into this new stuff, which I'll be looking at these calcium blips and this very kind of complicated process to open up a channel and what has to go right for that to happen. So let's talk about some of my previous work on this process that I call this diffusion with recharging traps. We've seen pictures like this. You can imagine that we have these particles in a domain diffusing or maybe moving according to, again, some general Markov process. And here are the interesting components I am placing on the boundary of this domain. And you can see here there are these two regions. We have these dashed black lines, which are going to be just purely absorbing. And then we have these red areas, which I'm calling these capture regions. And you can kind of think of them as receptors, in the sense that a particle could find an open available receptor. It could hit it, it will bind to it, and then some type of reaction has to take place. And then as a result, there's going to be some recharge time before that receptor can be available to capture again. So now we see that these particles are interacting with each other via the boundary condition, right? There's a couple different types of projects that you might have switching boundary conditions, but here I'm having that switch occur based on a particle hitting it, right? So now these particles are interacting with each other by changing this boundary condition. So, some of the initial questions that we had when we first kind of looked at this process was, okay, how many particles are going to be captured by these receptors versus escaping out of these absorbing conditions? What does the time course of, receptors bound look like? And things like that. The motivation in terms of, here I'm writing this kind of very general domain, but this was very much inspired by the biology that I was doing at the time. And that's specifically neuroscience. So you can imagine that we have the synapse, neurotransmitters released in the synaptic cleft, neurotransmitters diffuse out, some bind to that receptor, and then others are going to diffuse out. And I was interested at this time, because I was doing astrocyte research, how many of these neurotransmitters are going to diffuse out and interact with neighboring glia cells, astrocytes, microglia, and things like that. Right, so this was kind of the driving question, because I knew these glia cells were there, but it was kind of an open question of like, okay, well, how much are they actually doing in terms of recycling and things like that? And you can imagine that, okay, so we have this domain here, these glia cells are really good vacuum cleaners, essentially. So you can imagine that those are these nice absorbing regions. And then we have the receptors that are transmitting this neural signal that are acting like these recharging traps, right? So we kind of had this initial problem that was very much related to that kind of general setup that we had. So thinking about this diffusion with recharging traps led to kind of a series of papers. In our initial paper, we kind of showed that a ton of molecules are actually being essentially wasted in this process. So you can imagine that if there's no recharge time, you can essentially do a splitting probability type problem, and that's going to grow linearly in n, little n, where that's the number of particles released. But as soon as you have some type of recharge time, that now is going to drop to log n, as long as you have a large enough number of particles where that kind of outnumbers the number of receptors. And this was mentioned very much yesterday by Dennis in terms of, biology is redundant, right? It seems to be flooding the system. It doesn't need that many neurotransmitters in the system to actually pass on that signal. So it seems very much redundant, and there's a lot of this kind of washout activating potentially these outside astrocytes. More recently, I was kind of reading the experimental literature, and I found this work that was actually showing how these glia cells can very much ensheath, and how this ensheathment is actually time dependent. So this is an experimental figure here, where we actually have this glia cell in green ensheathing, inserting itself, and essentially turning off this inhibitory synapse via this ensheathment. So I was able to kind of revisit this microscale model. And in terms of this paper, we were able to essentially come up with an effective microscale model there, but we were really interested in kind of the neural dynamics, right? So we then derived this mean field neural theory that captures fine-grained correlations. This was originally going to be the core of my entire talk, was kind of going through this slide, but this has been published and it's out in the Bulletin of Math Biology. But I am happy to talk more about that to anyone's interested in the remaining time that we have here at this workshop. Another extension that we had with this was, me and Sean were kind of looking at this, and we updated this kind of Berg per cell problem. So you can imagine that we have a cell, it's sensing its environment, and they have this very nice classical result that you actually don't need a ton of receptors on the cell to have a good idea of saturating this maximal flux rate. But what happens if you have recharge? So we're able to kind of update this result. Here we were kind of going into continuum limit, homogenizing the boundary, and we're able to show that by having this finite recharge time, you can actually really strongly modify this classical result by Berg and Purcell. And again, more recently, in terms of work coming out of my lab, we've revisited this in terms of the blood-brain barrier, where we're seeing a similar thing in terms of molecules diffusing across the blood-brain barrier. And in this case, it's bidirectional transport, in which we have these amyloid beta proteins. It's a waste product that wants to be kind of flushed out of the brain and into the blood. And we're able to use this kind of continuum limit, extend it to have bidirectional transport, where again, we have this finite time. We have this ability to essentially saturate the receptors, and that leads to this massive concentration change between the vessel and the brain. And we're able to now modify that. There's a lot of work that shows that the blood-brain barrier actually changes during sleep. So we're able to do some simulations in terms of the REM cycle and how that helps with clearance. So I know that was quick, but that was just kind of to motivate how I think about these types of problems, right? I get inspired by some biology system that gives me a hard problem to work on. But then I like to revisit the biology and potentially make that problem even harder. But that allows me to have this tight connection with biology applications as I kind of advance this. And I also want to set this up because that's essentially what I'm doing now. I'm complicating this model even further by adding two components. I'm now going to essentially assume that these traps can capture multiple particles before it enters this recharge time. I'm also going to have these particles not just diffuse randomly but have a drift term there. Of course, in terms of that, a lot of the work that we've seen, we can extend that, right? The theory in terms of having some type of drift term there. It makes the math a lot harder, but we can do it. We can definitely use numerical methods to do that. But here I want to look to biology to actually consider a realistic drift term to see how that might affect some of the statistics. So with that, let's jump into this new process. So I'm going to take some time to talk about the biology. So there's going to be a couple slides here focusing on the biology, right? I want to be inspired by the biology to create this update to this diffusion with recharging traps. And in this case, the inspiration is coming from calcium. So calcium's everywhere. If you say you study calcium, that does not really limit the scope of what you're looking at. It's a tightly regulated process as a result because calcium is used for all these kind of downstream processes. So if you look in terms of a cell's concentration, it is very low. And what the cell has are these endoplasmic reticulums, calcium stores, so that when calcium is needed, it can essentially bind and release calcium into the cytoplasm. Again, this has been studied at length, both deterministically and stochastically. But in terms of kind of the new stuff are these very much different spatial scales. And this was actually highlighted earlier this week by James in terms of we have these blips, very short, we have these puffs, and we have these spikes of calcium activities. And I would say most of the literature has kind of focused on these spikes and puffs. And what I'm going to be looking at today is this single kind of this blip activity, which is kind of the shortest, fastest type of calcium response that we're seeing. Part of the things is that we're getting a lot more data that's able to have the temporal resolution as well as spatial resolution to look at these blips, and we're seeing them pop up in a lot more places. I mentioned that, I'm going to be looking, this calcium's coming from the ER, this IP3 receptor, and it's a complicated receptor. It has these 3 binding sites on it. It has one for IP3, it has one for activating calcium, and it has one for inhibiting calcium that kind of shuts off the channel. So here's some just more experimental data looking at these blips. Again, these blips are quite small. And then here you can also see, right, the time between these spikes can also be quite large, right? So this is where this kind of extreme statistics coming from in terms of the time between these calcium spikes. All these graphs are showing that these blips are most common when you have an IP3 receptor cluster of just one single IP3 receptor. So when you have just one, you're likely to see these kind of small blips appear in your experimental data. And I want to understand essentially the time between these blips, because that's the building block that then the blips turn into puffs and the puffs turn into spikes. So I want to understand kind of this basic building block first and foremost. So this is our driving question. How do we kind of get at the statistics of these blips, focusing specifically on kind of the time between them? As I mentioned, this IP3 receptor is kind of a mess. There's a lot of these binding sites here. Here's kind of the full kind of diagram. Out of all of these states, essentially this one in the upper right is in this activated state. So what is needed there? You need IP3 and you need the activated calcium site to be activated. To make things even more complicated is that there are 4 subunits on this receptor. I need 3 of the 4 subunits to be in this state for it to be open. So now we can think about this, right? Let's return back to this recharging trap. We now have these molecules diffusing in the cytoplasm. It's going to be looking for this small target. But now there's going to be multiple events that have to take place. Here, I'm not necessarily looking at the first passage time for a single calcium to find this target. What I need is 3 of them to find the target before the one that first arrives to unbind, right? That could very well be possible, right? One finds the target first. It takes so long for the next one that one actually unbinds and diffuses away. So in order for this channel to become open, I need essentially 3 of these calciums to bind. They need to be in this activated state for this channel to be open. I am going to simplify things for this talk. I'm going to essentially ignore IP3. I'm going to say that is either there or not there. And I'm not going to worry about the binding kinetics of IP3 for this talk. But here we already see that, okay, I now have a motivation to make my life harder in terms of I now have this motivation for traps that catch multiple particles before essentially recharging. In this case, it's this channel. So this is going to be opening up and causing these blips. All right, let's go into that next motivation. Like, right, I want to add some kind of non-trivial drift. And this has been kind of shown a lot recently. It's a little bit of an older result as well, but these ER, these endoplasmic reticulum, are known to actually have electrical signals. And with the outpouring of new data from these genetically encoded voltage indicators, so we can actually look at the fluorescence of voltage, this is actually popping up all over the place. The ER, presumably, it was kind of assumed to be essentially electrically passive, has been shown to actually have a pretty big potential difference between the ER and the cytoplasm. And that needs to be accounted for. What happens? Why do we care in this context that we have electrical signals in the ER? Well, as soon as you have a potential difference, you have to be careful because this thing pops up known as the Debye layer. So now you have an electrical field near the ER membrane. The ER membrane is acting as a capacitor. So there's a jump in potential. But then you have this Debye layer where we have an electrical field that's actually very non-trivial and changing very rapidly near the ER. Now, this is common in terms of neuroscience, in terms of the plasma membrane. It's typically ignored because it's usually on the order of a few nanometers, right? It is quite small. But think about what's happening here. In this case, the ER has basically this negative charge built up. And we're looking at calcium. Calcium has a positive charge. That means that this is going to actually help calcium kind of diffuse along the plasma membrane. So the question is, how does this fact that we have this Debye layer affect things like the ability for calcium to find these targets and bind? So, this is kind of a tricky problem to solve in its own right. How do you find this Debye layer? One thing that we have going for us is calcium actually doesn't participate largely in setting it. You can imagine that this calcium molecule, it is positively charged, but there's so many other ions in the cytoplasm, potassium, sodium, and chloride, that those are actually largely setting this layer. So imagine that I can essentially solve what is known as this Poisson-Nernst-Planck system to find this Debye layer, to find this electrical field, and then use that for my stochastic process. Again, there's a lot of details going into just this slide and finding this Debye layer, given the parameters that we know in the biology, to solve that. And I'm not going to focus on that here, because in this talk, I'm basically going to say, now I have this non-trivial drift. So instead of just looking at pure diffusion, I now have this particle diffusing as well as traveling along this electrical gradient. So we have this system here. You can imagine that, okay, well, how do I decide how many calcium molecules are actually diffusing in my domain? I can essentially assume that there's some bulk levels of calcium kind of known, some level of steady state. Like I said, it's small. There's a few kind of free calcium that are kind of diffusing in this space. So now I've made my life harder, but again, I've completed it. I'm not just adding, this because I want to, but because it is very much motivated by the underlying biology of the system. So altogether, I now have this movement for the particles in the space that depends on this background electrical field. I now have this trap that is going to be taking in multiple calcium molecules. And I have some state in which this channel will open where I need essentially this sum here, 3, right? I need 3 of these activating sites to be bound. I also don't need this inactivating site to be bound in the same spot, because that essentially cancels out that activated site. I'm going to assume that the boundary here is reflecting. And what I'm going to do is essentially add in this smaller reaction region around where these receptors are. So, if it's hitting the plasma membrane, it can definitely reflect off. As soon as it enters that reaction region, I assume that this reaction takes place with some right lambda. But it needs to be kind of in that proximity of the receptor. So this is the full system. And so now I'm going to go to the next part, because this full system, unfortunately, is not too helpful because of this Debye layer, right? We're talking on the order of nanometers. So if I want to simulate this system, it's actually quite difficult computationally speaking. Even if you get kind of creative with kind of a mesh where, near the plasma membrane, it gets really, much more dense than away. Really, because again, the time between these calcium events are on the order of seconds. So you have a nanometer spatial scale that you have to counteract with that. Again, that's kind of where this extreme events come from, right? Seconds doesn't sound like a lot, but when you have this very tiny spatial scale on top of that, that's where things get tricky. So let's update the question that I'm asking. I'm still interested in the time between these calcium blips. But now I'm specifically interested in how this is modulated by the surface charge caused by this capacitor that is the ER membrane, as well as how calcium levels in the bulk might be playing a role as well. And here, these details are what I've already mentioned in terms of this very computationally expensive. I have to become already clever a little bit to figure out how to deal with this. And the way that I'm going to do it is that I'm actually going to get rid of this, process, because there's a lot of this domain that nothing much is happening, right? Calcium is just kind of diffusing, right? The only interesting part are happening within these reaction regions. And that's where this approximation comes into play, where I'm now going to essentially look at this continuous time Markov process with discrete states, right? So now I'm looking at what's happening in this reaction region, which I'm calling omega R. Calcium can flow in. And once it's there, it can also bind to either the activating or inactivating site. So that's going to be kind of this binary 0, 1. And I can look at kind of the total number of activated sites, total number of inactivated sites within the reaction region. So what can happen with this Markov process? Well, we could have a calcium molecule wander into the reaction region. How fast does that happen? Well, I'm calling that lambda in. Once it's there, essentially, it can leave at some rate that I'm calling n times lambda out. To get a rough estimate for lambda out, I'm just going to solve a mean first passage time. And so here, right, we've seen this, right? We can do this calculation, but note that I have the background field here as well, right? This is not, we all know that we can make this more difficult. We are allowed to have a drift term here, but again, this is, been motivated by the underlying biology. And just assuming that we have kind of this uniform, I essentially estimate this mean first passage time for a particle to leave the domain, and I'm calling that lambda out. Once we have lambda out, I then essentially pick lambda in so that it essentially matches what I know to be in terms of the steady state concentration, which I'm calling N steady. This is going to be given by this Boltzmann equilibrium, which again accounts for this electrical field. What else can happen in this reaction region? Well, they can bind to either the activating site or the inactivating site. And that rate depends on how many sites are currently occupied. So I have to update that depending on, if there's already 2 activated sites, right? I have to update this lambda A to account for that. So I have this full system, this continuous time Markov chain, and I can simulate this process, right? Before, again, it was numerically very prohibitive. I can at least now simulate this process here. But I want to get greedy. I want to see is there any type of spatial scale or time scale that we can use to actually simplify this process a little bit more? Because you can do it. It's kind of a mess. There's still a lot of states to worry about. And it turns out that you can, because the rate of lambda out is actually much faster. What you end up having with, again, these parameters that are grounded in the biology of the system is that you end up having a lot of calcium molecules diffuse through the reaction region, nothing happens, and they just leave. So you actually have to have a couple of successful targets find the reaction region before binding even occurs. So I have these kind of inherent timescales of the system. Tau n is just that injection and leaving rate, independent of the reaction scheme, and tau s has to do with those reactions. And I see I have this epsilon term that has a separation of scales. And as a result, I can essentially do this quasi equilibrium here to say, okay, what is n? Fix s and sa and si. What is n as a result? So this is independent of state to leading order. So that's nice. So I'm able to significantly reduce my model. And I have just kind of this very much simplified model here. But if I think about this even a little bit more, I know, well, these sites, I'm going to assume that they're actually independent of one another. So yeah, so I have this continuous time Markov chain, but now I'm going to assume that these sites are actually independent. And I can now look at essentially what the probability, as that evolves in time. And I can look at kind of the probability that a particle is bound to a single activated site at this kind of steady state. And, this looks like this Michaelis-Menten form that I believe we're all familiar with. I can do that for the activated and inactivated sites. I can do here, if you notice, I have n here. I'm going to do, you just have to adjust units a little bit. And you can actually write this in terms of C, where C is now a concentration that's related to this Boltzmann distribution. And recall, right, what are we really interested in? We're interested in when this channel is open. We're interested in when this n value is greater than or equal to 3. Now we have the sum of 4 IID Bernoulli random variables. I'm able to push on that. I'm able to, get that full distribution for what n looks like. And now once I'm here, now I can get at exactly what I wanted to at the start, right? I want to look at this mean first passage time from closed to open, right? I want to look at the transition it takes. I'm in the closed state. How long does it take for me to open, right? This is that time between blips. So with this, we can write this formula down, right? It's the probability that we're in the closed state divided by this stationary flux from closed to open. And now, I've done, I made all these approximations, right? I started off by making my life harder, but then I've made these approximations and now make my life easier. And now this is just a textbook calculation to get this estimate for what this mean first passage time looks like. And so we can solve for both of these in closed forms. And so now we have an analytical formula at the end of the day for what this mean first passage time problem is. So now this is kind of the key result. So now I can do things and we'll ask, what does the field do to the time between blips? So the first line I want everyone to focus on is this red line. And notice that it looks pretty much monotonically decreasing. This is what we kind of expect, right? If there's not enough calcium in the system, it's going to take a really long time for calcium to find the receptor and to get into the activating state. As we increase calcium, right, we expect that time to go up. We expect more successes. As soon as I add the field, notice that I have this very strong non-monotonic behavior. I now have like this local minimum where, we have this initial decrease in the time between blips, but then as I increase calcium more, I actually get a slowdown. What's happening there? Well, that has to do with the activated site, right? If there's so much calcium in the system, there's more likely for calcium to be bound to the inactivating site, which is now going to actually slow up this process. The important thing to note is that in the field case, this is actually non-monotonic as well. If I extend calcium out to unrealistic values, this red curve actually extends out or pops back up. But this says that in the parameter regime that we're interested in, having this capacitor, this electrical field, really brings that non-monotonic nature into this relevant regime. So this is kind of our key result. As I mentioned, this is very much new work, but whenever you see kind of this nice non-monotonic behavior, that's already kind of a nice takeaway. So then I'll just kind of wrap up here by talking about a couple caveats and future work. Here, the mean first passage time, I'm starting in n equals 2. Basically, think about the channel was open and then initially closed. That means that n went from 3 to 2, right? So I'm not going from 0, none bound, to the open state. I'm assuming that I'm actually in this. I've recently closed, and I'm in that state. But when the channel's open, right, calcium is actually flowing through the channel, and I've kind of ignored that process, right? The fact that there is this high local calcium transient that happens when the channel's open. I've kind of ignored that, assume that it kind of gets washed out pretty quickly in terms of our domain. We have some simulations that look at now what's happening during the open time, right? What do the open statistics look like, and how does that depend on field? And again, we see actually a pretty significant influence here in terms of having the field versus no field. Here is a picture of an astrocyte, and imaging technology has improved it, in which we just don't have the star-like process. We actually have these very fine processes. So we actually have regions where the volume gets very small. So far, I've been assuming that I'm in this regime where I'm able to have this bulk calcium and the electrical field kind of levels off. But in reality, going back to the biology, I actually have not only the ER membrane to contend with, but the plasma membrane to contend with. And it's ongoing work in terms of accounting for this as well. So how does the geometry of this affect these statistics as well? The calcium is complicated. There's a lot of channels, so you can make it more realistic by including that and asking how that kind of has effect. If calcium gets high enough, my assumption that it doesn't affect the potential breaks down, so you have to be a little bit careful there. And then lastly, right, maybe this is enough. Maybe I want to just spend more time thinking about kind of this interesting kind of mathematical problem. Biology has served me well. Maybe there's a lot of interesting problems just to solve in now this updated framework. Where we have these searchers looking for these capture regions where multiple captures are needed to initiate some reaction, and where I have this non-trivial drift term. So with that, I want to thank my collaborators. A lot of this work was led by my PhD student, Tin Hao, and is also a collaborator at Minnesota, Hans Othmer. So those were the core work in terms of the calcium model that I talked about. The names in Gopher Brown here were collaborators with kind of the earlier stuff that I've worked on with the diffusion with recharging traps. So with that, thanks.
Questions?
Yeah.
Right.
The dark electric field are constant, or do you update it as the concentration changes?
I keep the field constant. Essentially, I'm assuming that the calcium is not impacting the electrical field. But I think that breaks down. As you go to these puffs and these waves, you have enough of accumulation of calcium in which now you have to update the electrical field. And that becomes even a harder problem. But right now in this work, I'm assuming that potassium, sodium chloride, that's setting the electrical field, and I can essentially keep that constant.
Thank you.
Fixed or constant? Fixed.
Fixed. Yeah, sorry, yeah, fixed. I came in fixed.
Yes.
I was wondering how the data is in terms of clustering of different groups of channels. Is it feasible to do that loss grading and keep going to get puffs and then waves?
That's what we hope. right? Our hope is that we can do some coarse graining. So, and our hope is that, right, we have to overcome this nanometer scale to get any hopes of puffs and waves, right? You have to look at larger space scale. You have to look at larger time scales. So we have to essentially use these statistics to do this kind of coarse grain so that we can change that. Yes.
Dennis?
If I remember correctly, you estimated the rate of arrival of calcium ions through the volume averaged mean first passage time.
Yes.
But this assumes that the ions diffuse everywhere, and
Yeah.
usually the calcium channels, they're quite close to the vesicles, and so in that case you have 3 orders of magnitude difference in time. They can go right directly in like in a few tenths of nanometers.
Because you're saying it's so crowded that it's not an open space. There are actually kind of these tunnels that are directed.
I wouldn't say that the volume of your domain is rather artificial, so it depends on what you take.
Yeah,
you'll get orders of magnitude difference.
For sure. No, and I think that kind of goes back to this, right? It depends on where are you looking at this calcium activity in the soma, in these processes, in the fine processes. I think that question very much depends. And this work is very much limited. And we were kind of thinking of it in kind of this intermediate region there. But 100%, the geometry is going to have a huge impact on these results. And I think that's one of the takeaways. Like, I think one of the things I'm nervous about with this is that anyone who's done a lot of calcium results was like, we've done this without worrying about the electrical potential for years, for decades. Why do we have to worry about it now? And I think part of my argument is it very much depends on the geometry. And we've been living in the soma. And now as we kind of get more data, I think we have to really think about the geometry to constrain our problems. I completely agree.
So you have adjacent receptors here, or is this a setup where you have?
Here I just have one single IP3 receptor.
Oh, just one. Okay.
But they have subunit.
Okay, so I am curious about this though. So once calcium particles attach or bind to the receptors, but then you have sort of multiple unbinding and rebinding events. Are those ones that made it there first? Do they tend to just like pop off and come back on? Like, have you looked at the statistics of how often those just like first there sort of are the ones that sort of get recycled, and the ones that didn't get there first, they just end up getting repelled and just end up sitting around in the bulk while this is going on?
Yeah, that's a good question. And we thought a lot about this, and part of this was essentially if you unbind, we're going to place you far enough away to prevent rebinding events like that. And there's some evidence that the receptor needs, again, maybe some time to recharge before just doing that. So we do that by placing it far enough away.
And do you think that there's clearance processes that might actually do that? Like,
yeah, because the other thing is there's a ton of calcium buffers, right? So if there's a calcium buffer there and you unbind, it's also possible that a calcium buffer comes and kind of sweeps you away as well. Yeah.
Yes. How many receptors are there, and what's their distribution?
Well, so it depends on the kind of what scale you are. So here, I'm just assuming that there's one with 4 subunits, and they're just right next to each other. But as you go up across these scales, again, you have these clusters that you can have high density, but you have these regions where these clusters could be kind of spread out. So it very much depends on the spatial scale you're looking at. But for this work, I assume I just have the one that are relatively close to one another. And I actually assume that once you're in the reaction region here, you're equally likely to bind to any of those.
So why didn't you use periodic boundary conditions? Why did you use no flux?
Oh, you can use periodic boundary conditions,
which changes things dramatically, right?
Yeah, you can. And we also thought very carefully about, what are the boundary conditions here. The main thing that we kind of want is that there's some baseline bulk calcium concentration away from this, and we want that to be fixed. How do you do that? You could have reflecting, you could have periodic, you could have absorbing, and then just inject new molecules, right? So there's a couple different ways to do it. And we did play around with that, and at least in terms of the results that we're showing here, it didn't depend too much on which one of those that we chose.
And the electric field it's really only working when you're close to the surface, right?
Yeah, in terms of having any effect on particle location, it is only having an impact when you're really close to the membrane. Yeah.
So there's some boundary layer analysis you could do there to try and turn it into just a purely bulk problem. And
Yep. No, for sure. I think there's a lot of interesting, yeah, and I think that's one I'm also very much tempted to look into as well.
any other questions? All right, thank you so much.